Luca Nicolini received his bachelor's degree in biology, and a few days ago a master's degree in molecular and cellular biology with a thesis on biotechnological applications of microbial communities associated with copepods. His main interests concern the microbiology and biotechnologies applied to the environment. And now I leave the floor to Luca Nicolini for his presentation concerning the study of the microbial communities associated with cultures of Acartia tonsa and involved in the degradation of polybutylene succinate-co-butylene adipate. You can start. Thank you.
Okay, thank you. Good afternoon, everyone. Today I'm going to explain to you results from collaborative studies conducted from the Department of Civil and Industrial Engineering and the Department of the Biology at the University of Pisa and the Italian Institutes for Environmental Protection and Research from Livorno. And the topic was to study the microbial communities associated with the culture of Copepoda, Calanoid Copepoda, Acartia tonsa, and involved in the degradation of PBSA. The aim of this work was to carry out a taxonomic and functional characterization of microbial communities associated with the culture of the marine Calanoid Copepoda, Acartia tonsa, reared under controlled conditions. A culture-independent approach was directed to the alive and dead adults of Acartia tonsa. Also, a culture-dependent approach was conducted to the Acartia tonsa culture debris, which we will see later. By integrating the culture-independent and culture-dependent approaches, our interest was directed to the hydrolytic activities associated with microbial communities potentially involved in the degradation. Sorry. Okay. Now, can you hear me now? Okay, sorry. By integrating culture-independent and culture-dependent approaches, our interest was directed to the hydrolytic activities potentially involved in the degradation of macromolecule recalcitrant to biodegradation. Especially, the interest was directed to the PBSA, which is a biopolymers which present some application in the marine environment, as we will see later, and present similar properties to some petroleum-derived plastics. So, what is Acartia tonsa? Acartia tonsa is a crustacean belonging to the Copepoda subclass, which is widely distributed in the marine environment. This species feeds mainly on phytoplankton, in particular on unicellular algae, filtering the water column. During the feedings, Acartia tonsa releases nutrients and it forms viscous water layers in their surrounding that favor a bacterial colonization from the water column. Those bacterial taxa can form then a zoosphere that is characterized by Copepoda bacteria transient nature interactions. Although the microbial communities associated with the copepods are influenced by their feeding habits, the evidence from the literature can show that there are some bacterial taxa that can form a stable association with the copepod and form in this manner a core microbiome. At the end of the Acartia tonsa life cycle associated with the carcasses formation, the microbial communities associated can utilize the macromolecule that constitutes the carcasses structure, which are proteins, lipids, and chitin. This is associated with a significant increase of the protease, lipase, and chitinase activities, which is potentially involved in the degradation of the macromolecule recalcitrant to biodegradation. With the deposition of carcasses, the microbial communities could meet anthropogenic origins material which is accumulated in the marine sediments. Starting from this observation, the hypothesis of this work is that the involvement of the enzymatic activities of the microbial community of the carcasses in the degradation of macromolecule recalcitrant to biodegradation, in particular biopolymers. In this thesis, we considered PBSA, which is a biopolymers that present some application in the marine environment, as we will see later. And for example, one of these application is the use of PBSA to the structuring of the nets for Posidonia oceanica replanting. What is PBSA? As you know, PBSA is formed, is a polyester that is formed by ester bonds, and they are susceptible to the carboxylic-ester hydrolases activities that can promote the hydrolysis of those bonds, and at the end of this process could restitute constituent monomers of this polymer. One important properties that regulate the degradation of the PBSA is the three-dimensional organization of the polymer chains that can be disposed in parallel to create a less accessible to the carboxyl-ester hydrolases activities regions, termed crystalline regions. The polymer chain can also form a supercoil that are more accessible to the same enzymatic activities and termed amorphous regions. So, in the experimental plan adopted in this work, as you can see, we started from the Acartia A.tonsa culture, reared from 2010 to until today in the ISPRA laboratories under controlled conditions. This allows us to verify our hypothesis relative to the hydrolytic activities associated to the microbial communities without any interference that come from natural microbial communities that you can find, for example, in the marine environment. So, at the beginning of these experiments, we reared Acartia A.tonsa with a monoalgal diet based on Rhinomonas reticulata, and after the acclimatization period, we considered Acartia A.tonsa culture to study the bacterial communities associated with the Copepoda that reached to the adult stage and those associated with the correspondent carcasses, adapting a culture-independent approach, which consists in the metabarcoding of the 16S rDNA associated with the reconstruction of their functional profiling. In particular, our interest was directed to the temporal dynamics of the bacterial communities associated with the alive adults, considering as experimental group the newly formed ones and those formed by 7 days. But especially the interest was directed to the carcasses, considering as experimental group the newly formed and those formed by 4 and 33 days. To equalize the starting condition of the bacterial communities associated from the carcasses, an artificially induced death was promoted. A culture-dependent approach was also adopted, and the interest was directed to study the interaction of the microbial communities with a microenvironment rich in organic substance, like this of the sedimentation of the Acartia A.tonsa carcasses, which is termed debris. And from this microenvironment, a bacterial and fungal morphotypes were isolated, and they shown the ability to use a PBSA as the only carbon source. So here are presented the results of the culture-independent approaches, and as you can see in the figure, the bacterial communities associated to the 33 days old carcasses were clearly separated from the other bacterial communities. For the beta diversity, multivariate statistical methods was applied, considering in particular weighted unifrac distance matrices calculated on our samples. As you here are represented the first two principal coordinates that could help to show more than 60% of the variance. So I know that this figure is a lot of they have a lot of complicated image, but as you can see, here are reported all the bacterial taxa that are associated with adults and death copepods, in particular of Acartia A.tonsa, and as you can see in the bracket are represented the relative abundance of each taxon, and in each cells are represented the standard score, which help with a color code to understand how much the relative abundance of each taxon differ from the average value calculated among all the experimental group. The bacterial communities in exams were represented mostly by the protobacteria, Bacteroidota and Actinobacteriota, as you see in this figure. And in the case of the bacterial communities associated from 33 days carcasses, you can see that they are represented by bacterial taxa other than protobacteria and Bacteroidota, allowing them to be clearly distinguished by the others bacterial communities, as you can see by the dendrograms here reported. In particular, for these communities, you can see two specific phyla that are Actinobacteriota and Firmicutes, which are described in literature as a fundamental part of core microbiome of Acartia A.tonsa. In the right figure, you can see the bacterial taxa at the family rank, and the principal observation is that the bacterial communities associated from the 7-day-olds alive adults are mostly represented by Flavobacteriaceae and Rhodobacteriaceae families, which are described in literature as a fundamental part of the copepod zoosphere, as I mean before. So, from the taxonomic data, we can reconstruct the functional profile of the bacterial communities, which is of our interest, focusing our attention in particular in the carboxyl-ester hydrolases activities, in particular in those described in literature in the degradation of the PBSA, which are carboxylesterase, triacylglycerol lipase, and chitinase activities. Here, as you can see, you can see the bacterial taxa that could help to show the most contribute to the carboxyl-ester hydrolase activities that are associated to the our bacterial communities. In particular, from the figure, could be highlighted that the bacterial communities associated with alive 7-day-olds alive adults, the contribution to the carboxyl-ester hydrolases is mostly represented by the Rhodobacteriaceae and the Flavobacteriaceae families. A different situation you can see in the bacterial communities associated of 33 days carcasses, and you can see that you have a marked equidistribution of the contribution to the carboxyl-ester hydrolase activities in the bacterial taxa that characterize them. And as you can see, these bacterial communities are mostly differentiated by the other bacterial communities, as you can see by the dendrogram here reported. In particular, this equidistribution can be evidenced for the actinobacteriota and in particular for the Mycobacteriaceae, Nocardioidaceae, and the Streptomycetaceae families, which are described in literature as involved in the degradation of cellulose and chitin, but also in the modification of several petroplastics. So here are presented the results of a culture-dependent approach. In particular, as I mean before, we isolated some bacterial taxa that carry out the capacity to utilize the PBSA as the only carbon source. One of them belonged to the genus Vibrio. In particular, we conducted a degradative experiment on PBSA granule, and here are reported the results of TIR ATR analysis conducted at the end of the incubation period. As you can see here, we have a shift of the peak of the crystalline regions from the uninoculated granule at the 1,715 centimeters at -1 to 1,707 centimeters and -1 in the uninoculated granule, especially at the end of the 82 days from the preparation of the inoculum. This means that we have a rearrangement of the polymer chain in the crystalline regions. And to understand if this rearrangement is could be associated to hydrolysis of ester bonds, we monitored the mass of the granules, but we didn't see any changes at the end of the incubation period. So we calculated the carbonyl index that could help to understand how many esteric bonds we can have in the PBSA structure. And as you can see here, you can see a significant reduction of the carbonyl index at the end of the incubation period, in particular 82 days, in case of the uninoculated granule compared to the uninoculated one. So this means that Vibrio can hydrolyze the esteric bond in PBSA and could help to degrade the PBSA. So just a few seconds. Okay. Among the fungal taxa that were isolated in our work, as you can see here, one of them belonged to the genus Cladosporium. And it shown the ability to degrade PBSA, not only at the surface level, but also in the whole PBSA granule mass. And this was associated to the significant reduction of the mass of the granule, starting from the 60 days from the preparation of the inoculum. Up to 92 days, as you can see there, we can see a further decrease of the mass of the granules, while we can see any significant reduction of the mass of the granule in case of the uninoculated ones. As the same situation for the Vibrio, in this case we conducted a TIR ATR analysis. In particular, here are represented the result from at the end of the period, in particular 27 days of the period of the incubations. And here you can see a shift of the peak relative to the amorphous region from 1,733 centimeters to -1 to 1,727 centimeters at -1 in case of the inoculated granule. And as the same situation for the Vibrio isolates, in this case we calculated the carbonyl index, in particular for the granule inoculated from the 60 days at the end of the incubation period. And as you can see there, we saw a significant reduction of the carbonyl index in the inoculated granule compared to the uninoculated ones. This means that Cladosporium could promote the degradation of the PBSA, in particular the hydrolysis of ester bonds. So, A culture-independent approach conducted us to bacterial communities that shown hydrolytic activities involved in the degradation of the PBSA. In particular, bacterial taxa associated with the alive adults are described in literature as forming a transient association with the copepod. But in the other end, as you can see, the bacterial communities associated with the carcasses are described in literature as forming stable associations with the copepod, in particular with the Acartia A.tonsa. And we saw that we have a marked equidistribution to the contribution of the PBSA degradation compared to the for the bacterial communities associated to the carcasses compared to the bacterial communities associated with the alive adults. In case of the culture-dependent approach, one of the bacterial isolates belong to the genus Vibrio, and it shown the capability to produce extracellular lipase that are involved in the hydrolysis of ester bonds in the PBSA, but just only at the surface level. In case of cladosporiums, as you can see here, it can produce a lipase that can not only degrade the PBSA at the superficial level, but also it can degrade the whole PBSA granule, and it was associated with the reduction of PBSA granule mass. So, to conclude, Ecopepod represent a nursery of microorganisms that show enzymatic activities of interest involved in the degradation of biopolymers with the potential application in the marine environment. In particular, in perspective, with the identification of the bacterial and fungal taxa that we isolated in this work, associated with their genome sequencing, we can obtain an entire metabolic pathway that maybe are involved in the degradation of the biopolymers. In particular setting, this kind of metabolic pathway could allow to standardize methods to apply specific application in the marine environment. One example is represented to the construction of the nets for Posidonia oceanica replanting, especially in the marine environment. Also, the bacterial and fungal isolates will be tested just on the modification of to the petroleum-derived plastics, and the refining of culture-dependent approaches will allow us to amplify our bacterial and fungal isolates that we will test not only in the PBSA degradation, but also in the biopolymer degradation experiments. To conclude, extending a culture-independent approaches to the fungal component, we will able to amplify the connaissance relative to the unexplored field, such as the fungal field, especially for the marine environment. So, thank you for your attention. I want to thank Patrizia Cinelli and Maurizio Seggiani to give me this occasion and to explain to you my work, and thank you for your patience. So.
Thank you very much, Luca, for your presentation.
You're welcome.
Are there any questions? Okay, Sita.
So very interesting, and I love the microbiology after a long time.
Yes, of course.
So between Vibrio and your fungal degradation, you see in one case it's surface, and the other one it's kind of bringing in bulk degradation.
Exactly.
And do you think the mycelium is entering the polymer, and therefore, and have you made any SEM analysis or anything to see what is the where is the bacteria or where is the fungus with respect to your polymer substrate?
So, thank you for your questions. We saw that during the degradation of the polymer, because we did an experiment directly in vitro, so you have your plates, and we put directly the granule directly on the agar medium. I don't know if you know the medium of from bacterial isolates or what else. And in case of the fungal isolates, we saw that the fungal structure goes directly inside the polymer structure, so it goes directly inside the granule and break all the structure. And in case of the bacterial isolates, in particular for Vibrio, we saw just only a sort of biofilm directly on the surface, just only this, without any
penetration.
Exactly,
Okay.
yes. Thank you for your questions.
Thank you.